this slide goes last, and it does, but I gave a similar talk, this talk, to ASU leadership that came a couple months ago. And I painstakingly put together facts, figures, all the science, all the data behind our strategic plan. And at the end of my talk, everyone came up to me and wanted to know the story behind this slide. Nobody asked me about the figures, nobody asked me about all the statistics I had done. They wanted to know what the story behind this slide was. So tonight I'm going to start with a little story so you guys get to know me and my background better. This is my son River. He was about 6 years old here, and he's swimming down to a massive star coral in the Bahamas. 6 years old, he was already one hell of a free diver. He was getting out like 30 feet. So, River grew up pretty much the same way I did. I was born and raised in Florida, a fourth-generation native, going all the way back to Florida Indian on my dad's side. I grew up around the ocean. My early childhood, I spent pretty much every day at the beach with my mom. Then on the weekends, I was hunting or fishing with my dad. I think I started surfing when I was 4, spearfishing shortly after. So I was a water kid. This is me at 6 years old. When I turned 6, this is my grandfather. I called him Pop. Me and Pop jumped on board his little 24-foot Columbia sailboat. We sailed down the west coast of Florida, throughout the Florida Keys, over to the Bahamas. We spent the whole summer. And I did this every year with Pop until I was 14. So every summer we would sail 2 or 3 months down the Keys, Bahamas, all around. And when he would call my parents to check in, he's like, I don't know how that boy's not waterlogged. He spends more time in the water than he does on the boat. But like any good grandparent, when we get to the Keys, he's like, where do you want to go? It's always up to me what our next destination was. So at 6, I was like his little navigator. But my answer was always the same. As soon as we would get to Marathon, I was like, I want to go to Carysfort Reef. So at the time, Carysfort Reef, which is off Key Largo, was one of the largest stands of elkhorn coral in the Caribbean. It was acre after acre, these massive, they look like antlers on an elk. There's all these channels, swim throughs, swim unders, and we would go there and I would just spend the entire days in the water. I mean, to me, it was endless excitement. I would come back to the boat, tell Pop the coolest thing that I saw, and be right back. So this was quite literally my childhood playground. When I started undergrad, I had already knew that I was probably going to become a coral scientist. And unfortunately, I had enjoyed this reef since childhood. All throughout high school, my friends and I would go down there and get lobsters, spearfishing, snorkeling, diving. But during undergrad, I got to witness the death of this reef, which is pretty traumatic for me. That's Carysfort Reef in 1975. In 85, it got hit in 83 by a minor bleaching event. And then in the mass global coral bleaching event of 97, 98, it pretty much took it out. And now this reef is a graveyard. Despite the millions and millions of dollars they put into restoration, it's not coming back. So this is an ecosystem that has been around for tens of thousands of years, gone in a relatively short period of time. And it's touching to me because, like I said, this was my childhood playground. So when I started grad school, I bought my own boat. I lived aboard for 14 years. Named Blue Healer after my Australian cattle dog, and I finished grad school living aboard. The harbor was so close to our molecular lab that I could pick up Wi-Fi from the cockpit of the boat. Finished my PhD, sailed down to Mexico, did a 3-year postdoc at UNO, and during that time I sailed a lot of the Western Caribbean. After I finished my postdoc, I sailed back to Florida and I started a sea school called Science Under Sail for undergrads. We would take 10 to 12 students aboard 2 to 3 week live aboard expeditions on sailing caravans. We would do the entire Zoom archipelago. It's like 710 islands. We'd start in Lutheran, we'd go all the way down to Georgetown. And we'd utilize a lot of my research sites from grad school and the postdoc to do surveys, different types of research, different types of experiments. And the students, not only did they get to learn marine science, but I taught them, I combined my two passions, sailing and science, and taught them seamanship, navigation, how to captain. So by the end of 2 weeks, they would take command of the vessel. It was basically their boat. They did everything on the boat, from captaining, navigating, dive ops, research, everything. So this program's graduated 53 alumni, and it was a transformative experience for all of them. And some of them have gone on to do careers and degrees in marine science. So when River got old enough, I started taking him. We'd sail my boat over for the beginning of summer, just loaded down with all the research gear. And then at the end of the summer, after the programs were over, he and I would sail up and down the islands for at least a few weeks. Sometimes we stayed 6 months. So he grew up kind of a lot like I did, and he came very, very connected to coral reefs. So in the Bahamas, our home port was in Georgetown. It's called Elizabeth Harbor, and this is one of the corals and reefs in Elizabeth Harbor. And I am very, very familiar with this coral. I've literally seen it hundreds of times throughout undergrad, PhD, and postdoc. I've taken genetic samples off this coral. I've done 3D photogrammetry models of this coral. I've done photophysiology on this coral. I even took a 36-inch core from this coral that gave us about 45 years of skeletal data that we were able to pull isotopic nitrogen out of those skeleton lines and demonstrate nutrient input into the harbor from the cruising sailors. Every winter, hundreds of boats overwinter in this harbor. And the data was enough to promote the park to provide a pump-out service for the sailing boats. So this coral was, pardon the humor, it was quite literally a rock star. 2 years ago it died. So this coral, I can make a pretty good estimate, was about 500 years old. So it was around when your great-great-great-great-grandparents were around. And for all of that time, it provided beauty, food, officers, and a place for kids of all ages to play. And it was gone in my son's lifetime. So I don't have any photos of the demise of that coral, but this is a coral I began monitoring in undergrad, and it's the same species, and it's about the same age. It's about 500 years old. This was 96, right before the 97-98 El Nino global massive coral bleaching event. Here it is in 97, completely bleached. 2000, there's a little bit of live tissue left. Got hit again in 2003, and now it's completely overrun with algae. So these are just two examples of corals that I'm familiar with, but this is happening all over the world. It's happening to reef systems everywhere. These bleaching events are increasing in frequency and severity. They used to be 10 years apart, and they're 4 years apart, and now it seems like the Great Barrier Reef State hit every year. Tons of headlines. The Great Barrier Reef is the largest biological structure on this planet. You're talking about an ecosystem that's been around for hundreds of thousands of years, and we're losing it in decades. So in Hawaii, this is America Samoa, but in Hawaii we got hit by the first marine heat wave in 2015. And then we got hit by another one in 2019. And the combination of these two, I'm told, reduced the coral cover by about 30% along the West Hawaii coast. That's a big deal. Corals are the foundational species for the reef. Without the corals and the three-dimensional structures that they create, nothing else is going to be there. So this was... Catlin Sea View survey did these 3 dimensionals. This was 2014, beautiful, healthy reef, staghorn coral. 2015, completely bleached, and this reef collapsed as well. So I'm glad that Time Magazine a couple years ago had the audacity to say that reefs are the most important place on Earth. I'm somewhat biased, but I can rattle on facts and figures about their importance all day long. And make no mistake, I'm a scientist, but first and foremost, I'm a dad. And I want these places, my playgrounds, to be around for my son's sons and his sons and daughters for generations to come. So given what we're facing, how do we do this? And I will get into our strategic plan now. I've been at restoration since it started, 25 years. I started my master's in the Florida Keys. It was some of the first coral reef restoration being done on vessel groundings. So ships would come up the Gulf Stream and try to hug the coast of Florida, and they'd get off course and they'd run aground. This was before global bleaching events. So we were doing restoration on acute impacts, and now we're frantically running around trying to figure out how to restore reefs that keep getting hit by these marine heat waves. So sexual production of corals engineered for restoration at consequential scales. So this is basically our strategic plan here for ʻākoʻakoʻa. And there's 2 things in this title that I want to highlight and that I will discuss. Corals engineered for restoration and consequential scales. So what do those 2 things mean? How do we engineer corals, and what's a consequential scale? So engineered for restoration, on the left is the IPCC scenarios for temperature increase over about the next 7,500 years. All of these scenarios are pretty much endgame for coral reefs. Coral reefs live within 1 to 2 degrees of their upper threshold. So a couple of degrees over the annual summer mean, and they start to bleach. So the 2 bottom scenarios, which are unlikely, there may be a chance for reefs, but the others, no chance whatsoever unless we do something. So I'll talk a little bit more about that. And consequential scales. This is the West Hawaii coastline. This is our area of restoration, and it's about 198 kilometers long. So the West Hawaii coast, this reef system, is longer than the circumference of Maui. It's obviously the largest reef system in the archipelago, and it's a huge area. So restoration has only been accomplished successfully around the world at meters, tens, maybe hundreds of meters, never at a scale like this. So how do we scale up restoration to have a positive consequence at this scale? So before I go further, I'm going to step back and just get into some basics of coral. These guys on the left, those are 2-week-old corals. And you can see the coloration in the top. That's their symbiotic algae that's incorrectly called zooxanthellae. That's the colloquial term that people know it by, but zooxanthellae refers to any brown alga, so it could refer to the diatom. But these guys associate with species in the family Symbiodiniaceae. This is a close-up of an adult coral, and you can see all this brown in there. That's high densities of alga that live intracellularly. They don't just live within the tissue, they actually live within the cell of the coral. So it's a very tight relationship. All reef-building corals have endosomites, and they can't live for very long without them. So the coral is an animal, but it's also a plant and mineral. So it's an animal that has an obligate relationship, that means it's necessary, with the alga, and then they form their skeleton. So the alga actually produce sugar and oxygen, which is food and fuel for the host, for the animal. And then the waste products of the animal, which are nitrogen and phosphorus, are food for the plant. That's why you find coral reefs in very eutrophic waters. They don't need the nutrients. They're photosynthetically powered organisms. So this is great, but when that relationship breaks down, then we have a problem. So this is a coral that's partially bleached. It is the alga that gives the coral most of its color. So when you hear the word coral bleaching, it doesn't mean that the coral is dead. It means that it's lost a high concentration of its somatic algae. So the left here is pretty bleached, and in the right, all that brown coloration are the algal cells. They occur 2 to 5 million cells per centimeter squared, so very high densities. You can see some of the fluorescence in the coral that's bleached. The coral produces a fluorescent pigments to try, it's like a sunscreen to try and protect the alga. So the results of elevated temperature, increased light are the same. When we have elevated temperatures or when a coral experiences too much irradiance, just like when you put your indoor house plant outside at noon in the sun, it basically fries the photosynthetic machinery. So at elevated temperatures or too much light, the target of bleaching is first the endosymbiont. The photosynthetic machinery gets overwhelmed and it starts transferring electrons to oxygen, creating reactive oxygen species. At that point in time, we still don't know who makes the decision, whether the coral says you're out of here or the alga's like, oh, it's getting hot in here, I'm out of here. But at that time, the association breaks down and there's this mass expulsion of alga. If the coral doesn't recover its alga within a couple of months, it essentially starves to death. So that's what happens after a mass bleaching event. If the temperature doesn't return to normal in a time frame that the coral can reacquire its alga, it basically starves to death. Time magazine called this nature's most beautiful death. So this reef looks beautiful, but it's heavily bleached. And all that coloration you see is the fluorescent pigments of the host. So the algae are gone. So it is beautiful. I've witnessed several mass bleaching events and it's gorgeous, but these reefs are on their way to collapse. So what can we do about this? Engineered for restoration at consequential scales. So let's talk about overcoming the challenge of scale. Up until about 10 years ago, most restoration was asexual fragmentation. So you take a piece of coral, you cut it into 2 pieces, and then you think that you have more coral, which you really do. So what we've done is we've figured out over the past decade how we reproduce corals in vitro. How do we bring them into the lab and make babies? So a coral that one in the middle is about the size of a volleyball. That coral will release about 1 million gametes when it spawns. So that's about 1 million opportunities to make new corals. The coral on the left is the cauliflower coral. It's producing sperm. Some of the corals are gonochoric. They have separate species. You can colony can be male or female. The Pastelopera, the cauliflower coral, it's hermaphroditic. So that colony will produce both eggs and sperm. So we figured out how to collect these in the lab and fertilize them. This is the first few rounds of cell division. We call it cleavage. So once we mix the gametes, we look at the embryos and we wait for cleavage. Once we get cleavage, we can estimate approximately how successful our fertilization was. So to overcome the challenge of scale, like I said, this volleyball-sized coral will produce about 1 million chances to make a baby coral. These are them after a day. They become competent, they start swimming around, and now they're ready to find a spot on the reef and settle down. So this is our coral nursery out there. It is currently the largest outdoor coral nursery in the world. So we back up to a volleyball-sized coral that can produce 1 million gametes. We've got 72 raceways. We put about 40 corals in a raceway. The numbers start getting really, really big. So this is what we are scaling up for. This year we have about 80 colonies of the 4 dominant reef building species that we're focused on. And we're going to try and produce 40 million larvae. And that's only using a few raceways. So over the next few years as we scale, all of these will be filled with brood stock. And it will be a massive operation to produce billions and billions of gametes and larvae and get them out there on the reef. So this happens naturally, right? Corals out in the field spawn. The recruitment is very low. Survivorship of corals is very, very low. So what we're implementing is a technique called targeted larval settlement. And this is the most impressive restoration that I've seen. There's been 2 studies conducted in the Philippines. They go down and they put these 5 by 5 meter mesh enclosures on the bottom. They build a frame, they put the enclosure, and then there's 2 divers dumping a bag of concentrated larvae in there. So by forcing them inside the enclosure to settle in high concentrations, survivorship is obviously higher than would naturally happen. And targeted, we have a very good understanding of all the hydrodynamics of all the reefs along Western Hawaii. So we can target where we put these new populations, such that in 3 to 5 years, when they become reproductively viable, we can kind of estimate where their larvae are going to go and settle. So obviously, if the prevailing current is this way, we wouldn't put all our targets down here to just get swept off the reef. We would put them up here. So that's how we're going to target larval settlement at high concentrations. The problem with this technique is it's very labor intensive. It takes 3 or 4 divers to go down and put that frame down, secure the mesh and all that. So when I got here, I wanted to come up with a way where we could scale this. How can we make a net system that we can deploy from boats? So I worked with a couple of the Kapuna. Uncle Damien, he's on our cultural advisory board, and his cousin, Uncle Paul, is a longtime fisherman. So they helped me with the first. This is the prototype, targeted larval settlement prototype net, and it's called polio, which is the Hawaiian word for jellyfish. Because when we launched this thing, the center of it sunk too rapidly, so I cut a pool noodle up and wrapped it around it as pink, and everybody was joking that it looked like a jellyfish, and it does. When we tried this first prototype, the problem with it was that the mesh is so fine that it would collect air, so it would constantly expand and then collapse on itself, so we weren't getting the breadth of the net that we wanted to. So Uncle Paul came along, and he's an impelling fisherman, and he's like, I think an impelling net would work just upside down. So this net has a rigid frame around it. So we built the second prototype, and much larger. It's 12-foot diameter, so I think it's 10.5 meters squared. And we spent 2 days hand stitching 140 weights around the perimeter and building this net to go out and test it. We tested it a couple weeks ago, and it worked perfectly. This is a long video, so you can see the net's got a frame, there's surface deployed. Uncle Paul says a good fisherman should be able to deploy about 20 of these in an hour. Settles on the bottom. And I dove down after we positioned the net, and it contours the bottom pretty well. I mean, there's a little bit of space, but it's acceptable. We experimented with polyvinyl alcohol bags. We were going to put our larval payload in the bag before we deployed the net, and then the bag would dissolve. But when we tested them, they get really gooey when they dissolve, and we're afraid that they'll trap the larvae. So this year, we're just going to have divers go down and put the larval payloads inside the nets. The nets stay on the reef for 2 to 3 days, and then we can retrieve them from the surface and use them again. Might have to do some minor repairs. We have 40 nets this year. We're going to try and deploy 40 million larvae within 40 nets at 2 different reef sites. If it works well, then we'll just keep scaling and scaling and scaling. So I created a logo that we're going to put on all the nets just to recognize their contribution to this, because I was thinking cast net, and I don't think we would have been successful without the input from Paul and from Vinny.
Is it okay to interrupt?
Yeah, go ahead.
I just had a question. How long would it take before you knew that it was successful?
So I'm talking successful on a large scale. So we will place the nets, leave them in place for 2 to 3 days, and then we pull the nets. We have divers go down and do what's called surface for motion. So they take a video camera and they swim a lawnmower grid, and then we can 3D model that reef area, and we can detect and map all the larvae in there. So even though they've settled, there's still going to be pretty high mortality over the first 9 months. So we will track them over 9 months and see what our outcome is at 9 months.
9 months.
And we also, this year, we're staining the larvae. We have 2 organic dyes, Nile blue and neutral red, which makes them bright blue or bright red and much easier to see. That dye will dissolve after about 3 months, but in the beginning, it'll allow us to quantify cells a lot better. So that's how we're going to attack the scales part. But what about the engineering part? If we just put a bunch of corals out there that are the same as the corals already out there, they're going to suffer the same fate. So corals reproduce, they put down larvae and recruits, then you get a temperature event, and the adult population suffer. So we are going to implement selective breeding. This has been done for tens of thousands of years in aquaculture successfully, and it's very simple and low tech. You have a corn plant over here that's drought tolerant, you have one over here that's pest resistant, you put them together, probably going to get offspring that are drought tolerant and pest resistant. So we're going for thermal tolerance. And we've done some of this in the Virgin Islands with good success. Most of the time, about 90% of the time, when you take 2 thermally tolerant parents, the offspring is thermally tolerant. The more you do that in backcross, you can increase that thermal tolerance. So if we can give these new corals, if we can engineer these babies that we're putting out there with the genes to withstand +2, +3 degrees Celsius, when they start reproducing, those genes will be spread throughout the ecosystem and will help kickstart natural recovery. So this is just an example. If you select the 2 tallest parents, your offspring are likely to be tall. So next year we will be implementing selective breeding. We've got hundreds of corals out there. We're setting up a thermal testing system right now. So we will be testing not only the parents to determine which broodstock that we want to do the selective process, but then when we do get our larval pools, we will pull from them and we will back test them. So we'll settle some of those larvae in the lab, rear them for a couple months, and then subject them to thermal tests to see, did we get what we want? Each round of that will guide the next round of selective process. So the goal is to produce billions of larvae that are engineered with thermal tolerance and be able to put them out there at a scale that has a consequence for a 198-kilometer reef system. So my vision is in a couple of years that fishermen are stopping by here, collecting their larval payload bags, taking them out where we want them. We see mass exodus of canoes and boats going out, putting larvae back out to the reef. Thank you.
It seems like you have a rigid base to your net, but the bottom of the ocean is not flat. So how do you get through the gaps?
So the rings, and they're just 2 pieces of fiberglass rebar. We bend into a circle. There's about 12-inch strings on those that hold the net. So that just keeps the net in its shape, but when it hits the bottom, they use a weighted line with fishing weights, and it contours the bottom pretty well. We also use a viewing glass, so if it does happen to fall like on a big coral and it's like that, we can just lift the net up and move it over a little bit. But I was impressed. We launched that net about 6 times. You know, and Uncle Paul would look down and he'd move it over if it wasn't sitting. But I go down every time and checked it, and I mean, maybe we had quarter-inch gap in some places, and that's acceptable. I mean, if some of the larvae get out, the fact that we can scale this allows for a little bit of loss, you know, versus sending divers down to do that complex free and all that. We're just going to get over that with numbers. Yes, sir.
So I have a basic question, actually 2 questions. First one is the polyp a coral or is the colony a coral, or both?
Oh.
Okay. I heard Ruth Cates talk about that.
Those little ones that I showed you that were 2 weeks old, once they settle, they put down a basal disc. So they start to produce calcium carbonate. And the first year of growth of a coral is mostly basal growth. We're trying to get secure. So when you get little corals, you get like, it's called spat.
Right.
So the first part is like several polyps, but it's flat. You won't get a branch or anything until later on because they need a solid foundation. But the polyp is the animal, yes.
And the second question is, to what degree are you working with DAR to get permission to do this?
So we have all of our permits now. DAR actually is doing restoration in the nursery. We have an agreement with them. They're doing reattachment work and then some asexual restoration. So fragmenting and stuff. But they go out with our teams and reattach large corals that are already reproductive, that have been blown off the reef or stones or whatever, and they'll go back and re-cement them so they can continue to reproduce in the field. We have all the permits. Sorry. The one thing that I want to implement, and I just wrote a huge argument to DAR and Brian Mason, is we want to do assisted gene flow. So we want to take corals, and we're not allowed to do that with adult corals because of potential pest migration. You know, if there's pests that are on the coral. But the larvae are essentially sterile. They're not sterile, but it's about as close as you can get. And there's been one population genetic study done in Hawaii on Brighties, the big boulder corals, and it's shown infrequent but connectivity between here and Maui. So if these things are connected intra-island, I can guarantee that it's happening along this coast. So to be able to take genes from the Levy and put them up north and assist that gene flow that would occur naturally anyway, that's going to be that's going to complement the selective breeding. Because what happens is when these populations fall below a certain threshold, there's not enough genetic diversity left for them to come back naturally. So we can assist that just by moving babies around.
Are you hybridizing to be more resistant to temperature?
Mm-hmm. Not between species, but within species.
Yeah.
Are you going to be doing this about cows?
This year, yes.
That's
Did you?
Because Uncle Damien helped me so much, our first selected site.
Do you have that research vessel that's out there right now? There's a research vessel out there today.
That's probably us.
Yeah. So the, I can't remember.
They're getting corals and looking for
I read
sex.
about it. What I'm wondering is, like in Honolulu, they have the closing community. Here's what's going on. They don't do that at Hope County, trying to do that at Hope County. But
I don't know that it's necessary. I mean, Honolulu is very shallow and a lot of tourists. We can't really do this type of deployment there because it's so shallow. We're thinking for next year that we just have snorkelers deploy the nets. But we're going to be doing, we have nine target sites up and down the coast.
so how long are those nets down there for?
2 to 3 days.
Are you gonna like close the zone off so people can't go in and mess with them?
They'll be labeled. We'll put stickers and placards on the floats asking people not to mess with them. They may still mess with them, but that's not all we can do.
Thank you. It's very interesting.
So it would seem that once you remove the nets, you've created a buffet, that you'll have localized predation.
But hopefully in 3 days, most of them have settled. So the fact that there's going to be a lot of mortality. So in the Philippines, at 9 months, survivorship was 0.0002. But at 40 million larvae, that means we put out 8,000 corals that survived to 9 months. They had 0 mortality after 9 months. So if you can get a coral to make it to 9 months, they're going to probably live. So we're going to overcome this scale thing by just sheer numbers. A lot of my past research was looking at ways to optimize fertilization, ways to optimize settlement using amino acid cues on the tiles. And right now, I don't care about any of that. I just want numbers.
So I didn't actually hear what 4 species you were going to put out there, but if one of them is cauliflower, then the crown of thorns are going to just collect, yeah?
We have put enough on them. I've only been on island a year, but I haven't seen nearly the crown thorns problem we had in Australia. Are they that bad?
The-
No.
It can be. Anywhere you find, there are locations where there are great populations that have recovered
Yeah.
from 10 years
I mean,
ago.
I've seen cots out here, but nothing like Australia.
Yeah.
You see armies of them just running across
Yeah.
the reef.
But you know, when the keiki are there, one cauliflower will take out a dozen, one cots will take out a dozen cauliflowers for breakfast. You know, the keiki cauliflowers.
Just numbers.
Yeah,
Put
okay.
enough out there and. Yes.
So FYI, most of the attention is to the Bay Area inside the Menehune Wall at Kahaluu. We've been diving, concentrating our efforts to look at coral recruitment, primarily cauliflower coral recruitment outside the wall, and it's amazing. I mean, it's really increased exponentially.
That's
So there are
really-
literally tens of thousands of new recruits there now, and I also monitor the cod predation. And I'm curious how many they can eat in a day or a month, or, I mean, I've seen data on the internet, and nothing seems to be too specific.
I'm not really sure.
And, you know, after they prey on cauliflower coral in particular, which seems to be their main source of food, you see the same effect as bleaching, and then within literally 4 to 6 weeks, they're fouled with algae, filamentous first, and they'll never be a coral again. It's a skeleton.
Luckily, the ʻākoʻakoʻa are fast growing. So
Yeah.
we get larvae out there, they reach maturity.
Do you know how fast?
Pretty quickly. The ʻākoʻakoʻa will become sexually mature in about 3 years.
We see like 1 to 2 inches per year of overall growth.
Varieties takes longer.
Yeah. The varieties don't seem to be affected as much.
Anything else?
So I have one other question that's only tangentially related to your presentation. And that's down at Honanau. There are some signs saying that there's pharmaceuticals in the water,
Yes.
and the signs have the ʻākoʻakoʻa logo on them. I'm wondering if you can tell me whether that's significant for any of the corals. In general, what you could tell me about that.
It is significant. We have chem catchers at several reefs that basically measure things like birth control. Ibuprofen is a big one, caffeine. And in other studies, ecotox studies, those do have a negative effect, not only on fish populations, but on corals.
There was caffeine and ibuprofen.
Ibuprofen and birth control are the main bad ones.
Okay.
There's a lot of caffeine, and the only way that gets in the water system is through us.
Yeah.
So, we're not doing any ecotox work right now, we're mainly focused on restoration, but, those are problems. So there's also sedimentation. So Greg, my boss, the director of the program, he's probably got one of the best data sets of any reef system in the world. So he does all the hyperspectral imagery, it flies over his airplane and so he's got 25 years of data, really good data on all this entire reef system. And now he's incorporating chem catchers. He can tell you places of high sedimentation, high nutrient influx, chemical influx. So he's working heavily with DAR because all those things can be fixed. Can we fix the climate change one? I don't know. With this administration, probably not. So that's why we're attacking this one because it's like a death by 1,000 needles. Those are all hurting the corals. You get a big marine heat wave, 30% in one blow. That's huge.
Hi, my name is Sue Ahrens, and I own Kona Coast Realty, but I also started Kona Ridge to Reef Institute. And for the past 30 years, I've been working with watersheds, floodways in North and South Kona, and also trying to get the county to upgrade the Kealokehe wastewater treatment plant from R2 to R1. Please help us to convince the mayor to make it a priority to stop 1.8 million gallons a day of R2 water going into Honokohau Harbor. That's what's happening now for the past 30 years. So that's really important. And also flood sediment, cesspools running into the ocean. The last main brown water advisory in Kailua Bay was just September of 2024. And we have to do ridge to reef.
Absolutely. Greg just published a pretty cool Nature paper showing that in the 2015 bleaching event, the reefs that weren't experiencing the other stressors fared better through the green heat wave. If we can take those regional stressors away, then reefs are better suited to
Handle that.
live through the next green heat wave, which is surely.
So I'm retired from here, spent 42 years for NOAA, but during 2014, 15, and again in 19, the temperature got up to 90 degrees at 60-foot depth. I know that from the water chemistry data that we take from our intake pipelines. So it's not just the surface, I mean, the few feet of surface water, it's a huge column of warm water.
And the double whammy is that it occurs during the summer. It's usually calmer, so you get less light attenuation. And synergistic excess light and temperature, that's negative feedback for the systems.
Why ASU? Why not Scripps or how did you guys become one of the leaders in the United
Thank
States?
you for asking that.
So I have a company called Coral Engineered Ecosystems. Greg hired my company to build a facility. I built a very large coral facility in St. Croix for the Nature Conservancy. I'm currently building one in Honduras for Roatan Marine Park in Cal Academy. So he hired me to build this, but I've also been research faculty working on this stuff for a long time. So in the 2-year process of building this facility, I got to chew his ear a little bit, because he was all ready to gear up and do just what TNC does, asexual fragmentation. And I convinced him that there was a better way, and showed him some of my work in the Virgin Islands doing sexual reproduction. So about 6 months before we commissioned the facility, he's like, hey, you want a faculty position? Oh, yeah.
So you're
I wasn't happy in St. Thomas. The university there is broken, and the island is just, it's not right. You're in a 26-mile long island, and you get 5 cruise ships every day. It's ridiculous. People complain when this one's in here, I'm like, oh my gosh, this is anybody else? Thank you.
Thank you, Robin.